Okay, rest portion. So we have described about the different end products formed during the fermentation. For the thermodynamic reasons, the bacteria that depend upon the fermentation process for growth cannot generate as much as energy as the aerobically respiring cells. In respiration, that means aerobic respiration, 38 ATP molecules can be generated as biologically useful energy from the complete oxidation of one molecule of glucose, which is assuming 1 NADH equal to 3 ATP molecule and 1 ATP is assuming from 1 ADP plus PA. Although 2 ATP molecules are generated by this glycolytic pathway, that is apparently enough energy to permit anaerobic growth of lactic acid bacteria and ethanolic fermenting yeast, for example Saccharomyces cerevisiae. The ATP synthesizing reactions in the glycolytic pathway, specifically it involves what? Substrate phosphorylation reactions catalyzed by phosphoglycerokinase and pyruvic kinase. Although all the ATP molecules which are available for the fermentative growth are believed to be generated by this substrate phosphorylation reaction, some energy equivalents are also generated by proton extrusion reactions also, which occurs in the intact membrane systems and are involved in the proto extrusion, which are applicable to fermentative metabolism. So the energy obtained by the bacterial fermentation of yeast. So comparing to the aerobic respiration, less energy is required by the fermentation of the bacteria comparing to the aerobic respiration. In this case, in the homolactic fermentation, here the glucose, as a result of the fermentation products, lactic acid is produced. Actual energy is 20,000 calorie per mole and the efficiency is 35. In the case of alcoholic fermentation, that is coming under heterolactic, here also ethanol is produced and here also actual energy 20,000 but the efficiency is 34. So these are all about the energy yield by the bacteria which are responsible for the fermentation by substrate phosphorylations. So these are all about brief description about fermentation. Mainly the fermentation can be divided into homolactic fermentation as well as heterolactic fermentation, etc. I think I have the notes of this fermentation. I will give you. You can check this PowerPoint also, and also I will send you some notes also. Okay. Thank you.